Okay, there it is. Well, hello, everybody. Um, I'm Burton Dickey and chair of the Department of Pulmonary Medicine at MD Anderson, and have done basic research for about 40 years, mostly in the area of vesicle traffic. And if you'll advance to the next slide, Jose. Um, this is a schematic of what we focused on for the past 20 years, which are innate immune defenses of the airway epithelium. Now, if you look at cells lined up in the middle, you see alternating ciliated and secretory cells. The ciliated cells are the ones with the little tufts of hair that propel mucus and particles and pathogens that we breathe in out of the lungs. And this cell mosaic covers the airways. Now, if you go up to the top line with the T1, T2, and T3, that indicates that immune reactions typically these days are divided into three types, type 1, 2, 3. And then if you go down to the very bottom, the blue lettering, you see what these different responses have evolved to defend against. And type 1 responses are primarily against intracellular microbes. Type 2 responses against parasites. These are helminths. And type 3 are against extracellular microbes. And then sort of moving up with the arrows, you may be familiar with the cytokines that are highly characteristic of each of these responses, with interferon in type 1, IL-13 in type 2, and IL-17 and 22 in the type 3 responses. And then what we primarily study are the arrows going out from the epithelium, which is how the epithelium defends itself against these different pathogens. Interferon responsive genes are the primary defense in type 1. The secretion of mucins and chitinases in type 2. And then the release of antimicrobial peptides and reactive oxygen in the type 3. Our primary focus is the synthesis and secretion of mucins into the airway. We take a mouse genetic approach, and we've identified many of the genes involved in the secretion of mucins. And we've also deleted both of the major mucin genes, MUC5AC and MUC5B, which gives us reagents to study. Currently, we're primarily focused on the biogenesis of mucin granules, looking at the subcellular trafficking through the Golgi apparatus. We've also done some work in type 3 immunity, stimulating the epithelium to defend itself against microbes like bacteria and viruses. And that has actually led to a practical application in the sense that we've learned how to stimulate the epithelium with an aerosol that results in a very powerful response that lasts for 3 to 5 days, and have founded a biotech company, and in an attempt to stimulate the epithelium in cancer patients, for example, who have lost their leukocytes during chemotherapy. And it's also undergoing a trial against COVID-19. So we're all about the sensing of pathogens by the epithelium and responding to that, and primarily focused on cell biological mechanisms. And we'd welcome either master's or PhD students into the lab. Thank you.